So welcome everyone, I'm Anna Petherick and I'm a Departmental lecturer in Public Policy at the Blavatnik School of Government. This conversation is part of a series that we're holding with experts about managing the response to COVID-19. And it is my pleasure to welcome Dr. Adam Ritchie, who is a senior project manager in vaccine development at the Jenner Institute, which is part of the University of Oxford. Welcome, Adam.
Hi Anna, thanks for inviting me along.
So I thought we'd start off with some basic science. So could you say in simple terms how the body musters an immune response to SARS-CoV-2, which is the virus that causes COVID-19?
Sure. So not surprisingly, given how new it is, we are a little bit light on specifics at the moment about some of this. Some of what we know is broad. How does the body make an immune response about any virus like this? Some of it is extrapolating from our understanding of related viruses, most notably the original SARS and also MERS, so severe acute respiratory syndrome and Middle East respiratory syndrome. And then the research has been carried out in the last couple of months on this. So I think one of the core things to consider with this virus is it is one that gets in, infects cells, and those cells tend to die in our body, and that induces an immune response. So that immune response can be things like antibodies, which you've probably heard about. They can be against the virus itself. There's great hope that they will be effective in providing long-term immunity, whether it's through vaccination or through natural exposure. But there's also other types of immune responses like the T-cell response, which is always very important for these sorts of viruses, because it is the T-cell response that can stop an infected cell from producing more viruses once it's become infected. So they're the main two arms I think we consider as being very important for this. Like its predecessor SARS, what we do know about SARS-CoV is that sometimes the immune response gets out of hand, induces something called a cytokine storm, and that cytokine storm can induce local and systemic inflammation throughout the body, and that can cause a lot of the problems that come with COVID-19 disease.
Thanks. So what do we know about how the strength of this immune response varies across different kinds of individuals?
So like most immune responses to most things, there is variability out there. There's been some concern that gets out in the press a little bit and in the public more widely, that perhaps the immune response against the virus is not very strong, or that there's a large number of people who don't appear to have an immune response. I think the data we're seeing at the moment suggests that's probably not true, but again, it's fairly early days. The great majority of people do appear to be having an immune response, including things like antibodies that have the potential to neutralize the virus and protect people from disease, or clear the virus once they've been infected. That strength of response is very different in different people. There's been reports that some people who have returned positive test results for the virus haven't been shown to then produce an immune response, but those reports are increasingly a minority of a population. That minority is getting smaller and smaller, it seems, from the data we've seen.
And just on those particular reports, from my reading, it seems that the question is probably about the veracity of the original test results, right? Is that your reading of it too?
So I think there's several questions about that. Tests don't usually get developed this fast. They usually tested a lot for, specificity, how much false positive, how much false negative are they going to produce? But these tests have gone into use very fast, which means there is the potential for error. So for instance, my expectation is the majority, if not all, the reports of people being reinfected is probably an indication of a test that actually gave a false negative, so they weren't actually uninfected at any point, or perhaps there was a false positive at one point that gave misinformation.
So let's talk about vaccines. There are currently hundreds of different vaccines in development, and this may seem like an obvious question, but what does this actually mean to have a vaccine from a public health point of view?
I think reasonably obviously, widely available, very effective vaccine alters the dynamic of this in many ways. It's one of the clear exit strategies for getting out of various levels of lockdown or social distancing or physical isolation, whatever you want to call it. But exactly how that's implemented and exactly what that means will vary jurisdiction to jurisdiction. I think one of the most important things to keep in mind when thinking about something like this is what having a vaccine actually means. So we're talking, people are talking about a vaccine in 12 to 18 months. The team here at Oxford, through our work, think it's actually plausible to have a vaccine this year. But there's a big difference between having a vaccine and having a vaccine available to everyone. And so the very early days may be something much more like having a vaccine that's available for emergency use, possibly. So for people who are very high risk and have underlying health conditions that make them at risk, both from infection and from severe morbidity and high risk of mortality. And then for those who are heavily exposed for professional reasons, such as frontline healthcare workers. So it may very well be that in the early days, having a vaccine means having a vaccine for some people, but not enough for everyone. But that can still have a big impact on the public health side, because having people protected who work on the front line makes it safer for them to go to work. It makes it easier for them to protect other people, and that can have a big impact. We saw during the Ebola outbreak that there were real issues around healthcare workers not being protected and how difficult that made their job.
So let's talk about how we get there. And there are many different strategies or technological platforms for developing vaccines.
Yeah.
Could you explain some of the main ones that are being used by different teams as they develop vaccines to SARS-CoV-2?
Sure. I think before I jump into answering that, I think one thing I would say that's important to keep in mind is we don't know what platform, what technology will produce the best results at this point. That's why there's so many different ones in development. We have our reasons for picking our one. Lots of other people have reasons for picking theirs, and people sometimes put it to us that there's a race against, other groups, other pharmaceutical companies to be the one who gets there first. To most of us, that's not what the race looks like. The race is just to get a vaccine or multiple vaccines available so that we can take things forward and get out of the sort of lockdown and risk situation. So having said that, there are lots of vaccines in development. There's high single figures now that have entered clinical trials. I think it's about 8 as of the other day. Hundreds are reported as being in development. There's different broad groups. Ours is a viral vectored vaccine. So specifically what we're called ChAdOx. Some people seem to be enjoying the fact our vaccine is called ChAd for some reason. ChAdOx is chimpanzee adenovirus Oxford. That's what the platform is. That's what that is we take another virus that can infect humans, but it doesn't cause disease. We alter it so that it's no longer able to replicate on its own, so that after someone's vaccinated, the viral vaccine doesn't replicate in them. And we put in part of whatever disease we're interested in vaccinating against. In this case, obviously COVID. So we put in the spike protein gene into our platform. There's lots of other viral vector platforms. There's direct genetic platforms. So where rather than inserting a virus or something like that that's carrying genetic material, you just insert genetic material that then gets the cells of the body to make a protein to induce an immune response. There's slightly more classical approaches that some people are looking at, like attenuated vaccines. That's where you take the actual disease causative agent and treat it in a way to make it not pathogenic. So you give it to someone, it induces an immune response, but it doesn't induce disease. And there's lots of current vaccines that are like that. So there's some of the main platforms that are being explored at the moment. There's something called the molecular clamp out of Queensland in Australia, which is another platform that's got potential here. There's all sorts of different technologies, but it's all about one thing. It's about delivering part of the SARS-CoV-2 virus to the immune system. So the immune system sees it, responds to it, so that if someone gets exposed later on, they've already got an immune response that's primed and ready to go.
So tell me about the tests that you guys have done so far and what else you need to do in terms of trials.
Yeah, so we currently have our phase 1 trial going in Oxford. That's fully recruited, I believe. And that's about 1,100 people in that trial, which is obviously very large for a phase 1 trial. It's primarily about safety. So all vaccines get first put into people to test that they don't cause harm.
And that's a phase 1 trial, just to be.
That's a classic phase 1 trial approach. So that's what we call our primary objective with this trial, to check that our vaccine doesn't cause harm. There's a second part of this trial which isn't really a classic phase 1 approach, which is to look for any signal of efficacy, which is why there's so many people in the trial. So about half the people in the trial have received the COVID vaccine, our Chadox vaccine, and half the people have received an unrelated vaccine that's used regularly in people. So that's the placebo group. It's what we call an active placebo, because these sorts of vaccines do have minor side effects, swelling in the arm, your immune response doing its job, sometimes fever, again, your immune response doing its job and responding to the vaccine. So if we had given, say, a saline injection, people would be able to know whether they'd gotten the vaccine or not, sometimes by whether or not they had any side effects. So that's where we are at the moment. There's plans for a larger phase 3 trial coming up. Those plans include expanding the groups. So the current trial is healthy adults with no underlying health conditions. Future trials will expand out to include special target groups, most notably, I think, in this context, the elderly, who, as most of us probably know, are at higher risk of COVID-19 disease and death. Okay.
So how long do these trials normally take, and how have they been expedited in the current context?
Yeah, so trials are part of a broader story about how long vaccine development takes, and some of the answer to this applies across the board to the whole process. A lot of the process is slow for non-scientific and non-medical reasons. So, for instance, there's normally quite a long delay between a phase 1 and a phase 2, or a phase 2 and a phase 3 trial. There's very long delays between preclinical data, developing the vaccine in the lab and taking it to clinical trial. There's then pretty long delays between clinical trial results and manufacturing. And the urgency of the current situation, what that's altered more than anything, is the delay element. So, for instance, just to take one very specific example, ethical approval for a clinical trial normally takes quite some time, not because it's difficult, but because there's always lots of trials going on and ethic review boards review them in order they come in. With the current COVID situation, a lot of the machinery around ethical trials, around ethics of trials and ethical approval, is focused on COVID, so that the people who are doing ethical reviews, who are independent from the study, do them in a matter of days rather than weeks or months. And it doesn't mean the review is any less rigorous. It just means they're focused on this particular review rather than other reviews. And that applies across the whole spectrum of this work.
Great. So let's talk about demonstrating efficacy. So when you test vaccines, you need a population that is going to naturally be exposed to the pathogen typically, or you've got to give the people in your study for efficacy usually a sort of a weakened form of the pathogen, and these are called challenge studies. So how are you going about testing efficacy, and are you kind of playing with when and where you're going to do these efficacy trials based on what's going on with the spread of the disease?
So this is a consideration. I've not heard any talk anywhere of challenge studies for this. So I think we can put that to the side, certainly for now. And I can't imagine Oxford being involved in anything like that, unless there was a clear therapeutic, like that would cure everyone 100% of the time. So challenge studies are only done in those situations where you know you can cure anyone who gets a disease. So what we're really looking at here is efficacy through exposure. So one of the reasons why the current trial is quite large is because when it was being planned a few months ago, because we have been working on this stuff for quite a while, well before lockdown and social isolation began, we were looking at when peak spread might be happening in the UK, and so it became clear that trying to get a decent sized trial quite quickly gave us a decent chance of getting an early efficacy result. But what you say is right. You know, we are looking at other regions, other areas, and I think personally, I think the most appropriate way to look at this is almost a scattergun sort of approach, which is to try and get trials started in as many places as possible, because it's difficult to predict with real accuracy where peaks of spread and exposure are going to be months in advance. So, you know, I know the clinical trial team, and I know some of our partners and potential partners in the biopharmaceutical industry are looking at trials across many countries at the moment, many different regions of the world. I mean, I think that also has the added benefit of I think it's quite important. This is a bit of a global enterprise rather than something that's just stuck in one place and centered in one area. So trying to get, you know, decent sized studies in multiple places where there might be spread happening gives us the best chance of seeing efficacy of the vaccine, but also efficacy in different populations, because populations do vary.
Great. So from a much broader perspective, an evolutionary perspective, if you vaccinate many people in a population, you're actually going to increase the evolutionary pressure on a virus to mutate. Have you guys been thinking about this? Have you been talking to evolutionary geneticists about this question?
Yeah, so it's certainly a question that's being discussed. I mean, I've not been talking to evolutionary biologists about this because my focus has been on getting the manufacturing system working. But it's certainly something that a lot of people are paying attention to. At this point, my view on what we've seen so far is there's nothing about this virus that suggests it's going to evolve quickly, sort of thing. So it may not be as responsive to that as some other things have been. So for instance, if we take something like malaria or something like HIV or something like the flu, they all evolve quite quickly, including under vaccine pressure, in the case particularly of malaria. Obviously, we don't have a good vaccine for HIV, so we've not seen if it would cause mutational pressure, but the expectation with HIV is that it really would very quickly. So it is something that lots of people are thinking about and paying attention to. The knowledge deficit here is possibly around related viruses. Obviously, in the case of SARS and MERS, both were contained reasonably early on, though we still see a little bit of MERS, but it tends to be quite limited in numbers. So the vaccines for those have not fully developed and have never been widely applied. So we've not seen if that has applied some sort of evolutionary selection pressure to those related viruses. So it is a little bit of an unknown, but there's nothing about it at the moment that suggests it's a particularly high risk. Now, if it does pan out, that is an issue. My primary expectation would be that's when we're likely to get into a situation we have with the flu, where we're looking at developing slightly altered vaccines every year or two, and then giving them to whether it's the whole population or just high-risk groups like we do with the flu. That is one potential approach to dealing with that issue if it arises.
Great. So let's talk about your home turf, academically speaking. So how do you make your vaccine, and what are the general challenges to mass production?
Sure. So we talked about all the different vaccines in development, and all have pros and cons. Ours, manufacturing-wise, can take a little bit longer than something like an RNA-based vaccine. So I'll walk you through that process a little bit. So as a little bit of background for this, I mean, people are a little bit surprised sometimes by how quickly Oxford has moved on getting a vaccine in place. There's two reasons for it. One is we have a team led by Sarah Gilbert who work on emerging pathogens. And so my colleagues, led by Teresa Lamb, who works with Sarah, developed the vaccine very fast because they're always working on any emerging pathogens. did Ebola work, they do MERS work. So all these emerging pathogens are right in their area. So that's step one, is we have made a vaccine fairly quickly. The other background is the group I work in, which is led by Sandy Douglas at the Jenner. He and I have been very focused on some of the economic issues around vaccines, primarily because we have an interest in rabies. So we have a Chadox against rabies that entered clinical trials late last year. That trial has now halted because of COVID, but we'll come back to that one day. But part of that process for the last few years, we've been working with some manufacturing experts, both in industry, but also academic ones we collaborate with, on trying to develop a process for making these sorts of vaccines that was scalable. So we do quite small batches in the lab, because we do in the lab for research, and then at a manufacturing site for small clinical trials. But there's a big difference between that and vaccinating millions of people. So we were already developing a process for the last few years around trying to have something that scales up to make millions of doses. We were planning to do that over the next couple of years. We just had to make it happen in a couple of months instead. So basically the way that process works is we grow up cells in a bioreactor. We put a pre-vaccine stock in with the cells. These are special cells which support replication of this virus. Remember, our Chadox vaccine doesn't replicate on its own. So these cells offer special support. It's able to replicate in those cells. A massive vaccine is produced. We harvest that out of the bioreactor, and then it goes through a series of processing steps where cell debris and materials from the original growth of the vaccine are all removed, and it's put into what's called a formulation buffer ultimately, which is what goes into a vial and one day will be injected into someone's arm. So in principle, the process scales. You can do it in a little bioreactor. You can do 3 litres. We're currently doing bioreactors at the 200 litre scale, and we've got plans to do bioreactors going up to 1,000 litre scale.
Okay, this leads me on to, I think, a really important question. So normally, when you develop a vaccine, you go through the steps to get it approved, and then if it is approved, you then go and make a lot of it for market. In this instance, do you think there's an argument for, or perhaps it should be, if you like, the norm, to mass produce before approval, so that we're ready to go?
Yeah, so I think there's no argument against it, is what I would say. The cost of manufacturing is relatively small. Now, they're big numbers compared to, our rent or what I get paid or something like that. But compared to the economic shock that is currently happening, they're really minuscule numbers. So Gates has talked about a couple of billion to take multiple vaccines forward. We think, tens of millions of pounds allows us to manufacture, tens of millions of doses of the vaccine in preparation for the results of the clinical trial coming through. Now, of course, if the clinical trial results show it's not a particularly effective vaccine, then those doses you could see as being wasted. But we don't know which vaccine is going to win. I think most governments have learned lessons over the recent decades that picking winners at the start is a very poor approach to things. So support for many candidates, at least a dozen, really is needed to take that forward. And if we spend 1 or 2 billion to get there, I think most people would think that's money well invested. The alternative is to wait, and that waiting, we are still on an accelerated timescale, so it's not a case of we'd wait 10 years, but you're probably talking about delaying by 6 to 12 months if we wait for clinical trial results to prepare manufacturing. What is the cost of that 6 to 12 months? I would say it's a lot more than a couple of billion US dollars.
Brilliant. I couldn't agree more. Dr. Adam Ritchie, thank you very much indeed.
That's all right. It's been my pleasure.